the layout of the suite of viruses, viral remnants, is exactly the same and could not happen unless it was inherited by a common ancestor. And so that's where we're at. So
ERVs, like when you do the phylogeny of ERV sequences, they nest within retroviral sequences. They have sequences like both the end regions and the genes that are only found in retroviruses, and they can actually be expressed in the human genome. Like some of the genes from these dead viruses can be expressed and cause problems. It's just all the evidence just says like they're from retroviruses, and that's the end of it.
Accordingly, retroviruses could have originated from within the host genome. Okay, you admitted that the host would have predated the virus. Okay, that's exactly what the variation-inducing genetic element hypothesis put forth by creationists suggests. Okay, accordingly, retroviruses could have originated from within the host genome instead of invading it from outside. This had to have been the case in any scenario since viruses are dependent upon host cells. This means that the cell's genome had to predate the viruses. You can read all this yourself.
So going back to the question I asked, is there a way to differentiate a virus mimic and a legitimate ERV?
Right, so that's a good question. I would say the majority of what we're looking at in terms of these, quote unquote genomic fossils that evolutionists such as yourself look to and compare across different groups, different species that fall within a nested hierarchical pattern, as well as the mutations that occur in these LTRs, these LTR elements. I would say that the vast majority, if not all of those, are created units of DNA function. Endogenous Retrovirus Handbook. So for anybody who is interested, this is a topic that is frequently discussed these days. You can get the full color version. Benjamin did a fantastic job. Probably the most comprehensive book you're going to find on the market on this specific topic, black and white version. And you won't regret it. You won't regret it.
All right, and we are live. Hey everybody, this is Matt Powell from Dinosaur Adventureland. I'm here with Donnie from Standing for Truth, and we're going to hear about endogenous retroviruses today, and I'm super excited about it because this is supposedly the best evidence for evolution. This is supposedly their best argument against us, that a lot of them will say. So I think let's just dive right into the presentation. Donnie, tell us a little bit about your channel, and thank you so much for coming on.
Appreciate it, Matt. I'm looking forward to this, brother. Yes, the evolutionary community, these days especially, they look to endogenous retroviruses as supposedly, the number one line of evidence for evolution. So in my slideshow presentation, I've got a few pictures and things like that in terms of our ministry. So I'll definitely give the audience some details there when I get my presentation going. So Matt, thank you for hosting this, brother. I'm looking forward to it.
No problem. Thank you for being willing to present.
All right. Well, you know what? If that's, I'll get my screen shared and we'll kind of get right into it, Matt.
All right. I'll have my mic muted.
Okay.
And so that way it'll reduce any background sound.
if there's any issues, you just let me know. My screen will be shared, so I won't really see anything other than my presentation. So you just let me know how everything's going. Okay, I just clicked screen share, so I guess once you let me know from my end if it's good, I'll get the presentation going.
1 second, I gotta go change one thing.
No worries. I see people coming in the live chat, so okay. Perfect. I'll shoot the link out.
Okay, it should be all set now, I think.
All right, perfect. I'll get my slides up, and then you just let me know if you can see it from your end. I'll click hide here, and now at this point, this is all I can see is the slides. So as long as the sound and everything's coming in.
Everything is good, my man.
Okay, brother. Appreciate you. Appreciate this. Okay, let's get going. So my presentation tonight, as Brother Matt put it, will be on endogenous retroviruses, ancient viral infections, or created units of DNA function. My name is Donny B. I'm married. I got 3 kids, and I am just fascinated with the topic of origins and ancestry. So endogenous retroviruses in general are small pieces of DNA that are found within the genomes of many creatures, many kinds of creatures, I should say. Now, the question is, are these DNA elements the ancient remnants of past viral infections, as the opening slide here says, or created units of DNA function? This is the most important question, and this question will be answered here in this presentation. I've written a very comprehensive book on this, the Endogenous Retrovirus Handbook, Dismantling the Best Evidence for Common Descent. I've got it right here that I can show the audience. And in this book, I answer, the toughest questions that the evolutionary community has put forth. So the reason why I wrote this book was because of the fact that the evolutionists, especially the ones that we've been debating in our Evolution Debate Challenge series, and I've been studying this topic for a few years now, they've been putting forth this argument from endogenous retroviruses nearly every single debate. If you YouTube, best evidence for evolution, you'll get several videos that come up as endogenous retroviruses. So I do highly recommend this book, especially for a comprehensive explanation and answer to all of their best questions, objections, arguments. So it is available now. You can get it through Amazon, and the orange version is the limited edition version that I've been getting a lot of good feedback on this. So that'll be up for a little while before I put it back to the normal version, which you can see here. There's a color version and a black and white. So you can also get it through our website, which is right here, Stand for Truth, ministries.com. And if you go to the shop icon, that'll take you to creationistclothing.com. So that's where you can find all our books. As a team, there are several of us. So I've got Matt Naylor. He runs the ministry with me. And we pretty much spend all day brainstorming and chatting about creation, evolution, and doing projects together behind the scenes. Our team geologist is Professor David McQueen. He used to teach at ICR, Institute for Creation Research. He's done a number of debates for the ministry as well. And he has his own book that we sell through the website. And then George Bond. He's also involved in research for the ministry in terms of flood geology. And of course, we've got a lot of behind the scenes guys that help with our designs, thumbnails, so on and so forth. So I'm just going to get right into it. When we talk about evolution, the question is, what do we mean by evolution? And is evolution something that young earth creationists, biblical creationists reject? Okay, well, the answer to that is no. So when we ask the question, are endogenous retroviruses good evidence for evolution? Well, it depends what you mean by evolution. A basic definition of evolution, especially when it comes to biological evolution, is changes in allele frequencies in populations over generations or over time. Okay, nobody's going to disagree with that. Yes, Change happens. We see a great variety of change, for example, in domestic dogs. Okay, so change is definitely something we see. As a matter of fact, biblical creationists, they don't hold to what's called the fixity of species. No, we don't believe that species or genomes are static. We believe they are ever-changing. And ever-changing environments require ever-changing genomes. But the problem is, the type of allele frequency changes that we see, they are not the type of vertical changes that would be necessary to explain what? To explain the fairy tale. The fairy tale of fish to fisherman evolution, bacteria to biologist evolution, pond scum to people evolution. Okay, which says that not only are wolves, coyotes, and domestic dogs related, which I would say, yes, that I agree with. That's scientific. But if you say domestic dogs, wolves, banana plants, pine trees, and whales are related, well, that's where we're going to have a problem. That's the type of evolution that's not scientific. Now, what biblical creationists hold to is what's called separate ancestry or independent origins. The evolutionists, they believe in universal common ancestry. So they say whales and pine trees are related. We just had a debate. How are whales and pine trees related? And the evolutionist typically answers, well, they're both eukaryotes. Okay, so they believe in universal common ancestry. We believe in separate ancestry, that God would have at the beginning created distinct, unique, separate created kinds, independent of each other. So God would have created, as he said in Genesis, right? The Bible claims to be the history book of the universe. And the Bible says that God created two people, Adam and Eve, in the beginning. Completely separate, this is where we get separate ancestry, from all other forms of life. Completely separate from your orangutans, separate from your chimpanzees, separate from, believe it or not, your pine trees. Now, these original created kinds, God would have front-loaded with what's called created heterozygosity. Now, that's just a fancy way of saying design diversity, the design diversity hypothesis. God said, be fruitful and multiply. He didn't say to be fruitful and clone yourselves. So it makes sense both scientifically and theologically for God to have created Adam and Eve and the original created kinds with pre-existing functional DNA differences. And through recombination, which is just the shuffling of these DNA variants every single generation, and other genetic processes, we can get change almost immediately, since those DNA differences are built in from the start. So we do believe in change, and this design diversity hypothesis, as people are going to see, it means that there's a great amount of morphological adaptability built into each kind. Now, these changes, they're going to be horizontal or oftentimes downhill. Species are going to hit walls. While the evolutionary model would require large-scale changes, increases in complexity, and net information. Where the creationists, we look to the beginning of time, we look to the expansion of genetic information. We're losing genetic information and variability over time. Well, the evolutionist looks back in time to the contraction of genetic information. So here on the screen right here is a picture of what you could imagine the separate ancestry model is like. And this is from the book Contested Bones by Christopher Roop and Dr. John Sanford. Highly recommend this book. And this demonstrates that, yes, we can, what some creationists probably understand as the phylogeny challenge. We have been able to, through mitochondrial DNA, Y chromosome, DNA function, we have been able to determine that humans are separately created from every other form of life, just like the Bible says. And the details on this will be left for another presentation. So endogenous retroviruses are not evidence for common descent. Let's go over some of the details in terms of endogenous retroviruses. So what are they? According to the standard...
Hey, Donny.
Yes.
Hey, sorry to interrupt. I'm going to go monitor the chat because I see some stuff going on in the chat here. So I'll be off camera for this.
Okay.
Do you want to take questions afterwards or just do the presentation?
It's up to you. I know you guys usually go live with your Bible study, so we can do the presentation and then wrap it up if that works for you.
Right on. All right. Hey, sounds good. So here's what we'll do, folks. Donnie's going to continue his presentation. If you have any good questions to ask him, any sound questions, make sure to put them in the chat. and I'll try to get to them, and then we'll have him answer those questions for you at the very end. And so, Donnie, go ahead and continue, and I'll be monitoring the chat.
Okay. Appreciate it, brother. All right, so let's get right into this. Endogenous retroviruses, what are they? And how this applies to design diversity is the fact that we would predict that based on the design diversity starting point, or again, created heterozygosity, that's just a fancy way of saying a state of what? DNA diversity, which makes sense of God's command to be fruitful and multiply. Okay, so what this means is God would have also front-loaded the genome with functional and beneficial DNA elements. And this is where endogenous retroviruses come into play. Because if what we as biblical creationists are putting forth here is true, then we should expect DNA function all throughout the genome. So endogenous retroviruses, what are they? According to the standard evolutionary story of endogenous retroviruses, an ERV or ERV is a stretch of DNA found in your DNA that got there when one of your ancestors was infected by a retrovirus. Now, one thing I do all throughout the book right here is cite a secular source for everything I say, because little do the evolutionists know, their very own scientific literature refutes endogenous retroviruses as evidence for evolution. So proponents of common ancestry believe that ERV elements are ancient remnants of past viral infections. Some evolutionists will refer to ERV sequences as fossil viruses. And you'll notice the source here, demystified human endogenous retroviruses. Human endogenous retroviruses, or HERVs. So when you see HERVs, so you see an H before the ERV, that's just representing a human specific endogenous retrovirus, or a taxonomically restricted retrovirus. Represent footprints of previous retroviral infection and have been termed, again, fossil viruses. Evolutionists acknowledge that for most of these ERV sequences, the infections would have occurred millions of years ago. So even the evolutionary community will admit, yes, we see these DNA elements in the genomes of living organisms. Did we necessarily see them get there? No, these infections would have occurred millions of years ago. So there is a built-in assumption there right away, as you can tell. Relics of exogenous retroviral infections. So exogenous retroviruses, most people will probably understand the HIV virus. That's an exogenous retrovirus. When you hear endogenous retrovirus, that's because it's occurring from within. It's originating from within. So ERVs are relics of exogenous retroviral infections of germ cells that result in integration of proviral DNA into the host genome. So the evolutionists, they assume that millions of years ago, these exogenous retroviruses from outside infiltrated the genomes of living organisms, and infiltrated in such a way where they could be passed on vertically through the germ cell lines and then become fixed in the genome. And so every single cell now has this ERV element, essentially. Now, the structure of ERVs or ERVs match modern retroviruses, as I pointed out, for example, HIV. On both ends of the retroviral DNA will be two identical sequences known as the LTRs, as you can see here in the diagram. That stands for long terminal repeats. So you got the LTRs, the GAG, the POL, and the ENV genes, they're called, or elements. In between the LTRs, we find, again, the GAG, the POL. The POL codes for what's called the reverse transcriptase. And reverse transcriptase is essentially just transcription, right? DNA, RNA, protein, okay? DNA transcribed in RNA, translated into protein. It's just transcription process in reverse. And that's necessary for the exogenous retrovirus because it has RNA as its genetic material. And so it needs to implement its RNA into DNA form. So transcription in reverse. Now, in between, or with the ENV, that stands for envelope protein, and that codes for the envelope that makes up the body of the virus. And these structures are common in ERVs and exogenous retroviruses. So this is essentially where the evolutionary argument comes into play. Okay, so here's some important questions that we're going to answer in this presentation. Remember, ERVs are assumed to be the ancient remnants of past viral infections. They've admitted this in the technical literature, as I've demonstrated. Evolutionists consider these to be genetic fossils that point us to common descent. Advocates of common ancestry would say that the chances of two ERVs being inserted, you'll hear this over and over again in debates and in their videos. Their main argument is, well, the chances of two ERVs being inserted at the same exact location in separate organisms are very small. They will argue that the chance of a human and a chimpanzee being infected in the exact same spot by the same specific type of virus is less than 1 in 10 million. And to them, this is highly unlikely, of course. The more shared ERV sequences, the more unlikely it becomes that these were inserted independently, they'll say. But remember, that's based on this assumption that they were inserted and they actually took place millions of years ago through exogenous retroviral-like infections. But if these ERV sequences really are created units of DNA function, then this whole probability argument completely falls apart. So here's some very specific questions that we're going to answer. Why are there ERV sequences shared between the genomes of organisms? If viral-like sequences in the genomes of organisms are functional, why do they bear similarity to a viral genetic material, right? The GAG, the POL, the LTRs on either sides, the ENV, and we're going to answer that. What is the best explanation for the nested hierarchical distribution of endogenous retroviral-like sequences in primate genomes? Now, what that means, for anybody wondering, that's if they're not familiar with a nested hierarchy, well, the elements, they fall into a nested hierarchical distribution, the ERV elements, that is, where more ERV-like sequences are found in similar locations between a human and a chimpanzee. Okay, these groups within groups patterns. And with fewer ERV-like sequences shared between what is assumed to be a more distant relative by the evolutionary community. Of course, we don't believe we're apes. We don't believe we're related to apes. This is what the evolutionist assumes, okay? Such as a gorilla or orangutan, a bonobo, lemur, so on and so forth. So evolutionary scientists appeal to what is called a family tree, and they interpret the nested hierarchical patterns within primate genomes, okay, in terms of their arrangement, as in, humans share more with chimpanzees than humans do with an orangutan. Well, they interpret this as reflecting common ancestry, large-scale evolution. And a big difference there is proponents of evolution, they believe in universal common ancestry, as I pointed out in the beginning, which is why this is important to set the foundation. So they'll say all primates are related, while we as biblical creationists, we say there exists separate ancestry. So in a nutshell, and just the easiest way to explain it, overall, in terms of anatomy, morphology, physiology, and genetics, humans and chimpanzees share more, which just standing back and looking at the organism shouldn't be a surprise, than humans would with a dog or a fish. Okay, so the next question will be, can the separate ancestry model explain the data better than the common descent model? And the answer is yes. The answer is yes. And we're going to start with the overturning of junk DNA. Because remember, the evolutionary community, they've always had to assume that the vast majority of our genome is based on genomic fossils, evolutionary leftovers, and junk DNA. And they invoke this because they have a deep time history, millions of years of evolutionary change, the buildup of what they assume are neutral mutations over time, which we understand has been completely overturned. No mutation is absolutely neutral in the effect that it has no consequence on fitness. We now understand that most mutations are nearly neutral, and that's actually detrimental to the neutral theory of evolution here. So they have to assume that the majority of the genome is junk in order for one, mutation accumulation to not drive us into quick extinction. Okay, so now what we actually understand is that most of the genome has tremendous, incredible evidence for biochemical function or activity. It was in 2012 that the ENCODE project came out with remarkable findings indicating that upward of 80% of the human genome was active to some extent, not predicted by the evolutionary community. You'll notice, and I've had many debates with these guys, PhD scientists, serious students of evolution, so on and so forth, and you'll ask them, did you predict DNA function in these ERV sequences prior to the discovery of them? And they'll admit no. So they've retrofitted the data, okay, as we're going to see as we move on here. So this ENCODE project, it came out with remarkable findings indicating that upwards of 80%, let me repeat that, 80% of the human genome was active to some extent. And that activity would be interpreted as what's called detectable biochemical function. Now, it was surprising that the genome consists of over 20,000 genes. This was an estimate. And this was not an anticipated number. Again, this wasn't predicted. This wasn't anticipated. What we now know is that a vast portion of the human genome consists of non-coding DNA sequences, which just means that these are sequences that don't directly code for protein products. Remember, you got DNA, RNA, protein. So in this case, you have DNA to RNA, and then those RNA products are used for a variety of amazing functions rather than directly coding for a protein. So this is a major reason why so many in the evolutionary community assume that most of the human genome constituted useless evolutionary baggage. And most evolutionists today will still defend this position of junk DNA. So we've discovered just innumerable functions of the so-called junk DNA. It's been completely overturned. There's what's called the nucleoskeletal hypothesis, which the details on that could be saved for a separate presentation. They act as a mutational buffer, gene regulation, expression, embryological development, maintaining the 3D structure of the genome itself. Pseudogenes, one of the evolutionists' favorite arguments as well, now known to be necessary to sustain healthy life processes in the cell. Gene regulation, they act as a decoy. Cell stress responses, gene regulation again. The ERV sequences, we now know, play an antiviral function. They're involved in tumor suppression, gene regulation. So here's a quick slide from an article by Dr. Rob Carter. And he points out one of the big revelations of the Human Genome Project was that genes that are used together do not necessarily appear near one another in the genome. Claims like it's just junk and the genome is nothing more than millions of years of genetic accidents were raised. This did not last long, however. For once people started looking into how the genome is organized in the nucleus, they realized that not only does each chromosome have a specified position in the nucleus, but genes that are used together are generally found next to each other in 3D space, even when they are found on different chromosomes. So we have genes interaction, and what's going on the molecular level appears to be science fiction, and it's function after function. It's mind-boggling. As Brother Kent Hovind likes to point out, you know, one single cell is more complicated than the entire space shuttle. And here's just a number of technical papers from the scientific literature demonstrating exactly what I'm saying in terms of the overturning of junk DNA. So in my book, it's very heavy on technical sources backing up everything that I'm saying. So none of this is even creationist literature. So the evolutionists can't complain. You know, we're destroying them with creationist literature, evolutionary literature, and it's just not looking good for them. You're going to hear me talk a lot about gene regulation. And so when you hear me talk about gene regulation, what that is it's how a cell controls which genes out of the many genes in its genome are turned on. It turns out they have millions and millions of genetic switches in our genome that are just waiting to be turned on or off via environment, which is evidence of what's called forward thinking, which points us back to the forward thinker. Okay, we have what's called a lot of functional redundancy. Picture how car manufacturers design vehicles with a spare tire. To the evolutionist, that spare tire is junk, it's useless, it's redundant. Why is it there? Well, if you're driving in a harsh area, or it's snowing, it's raining, whatever, and you get a flat tire. You drive over something, you get a flat tire. Well, you know what? That spare tire is going to come in handy. And we have functional redundancy like that all throughout the genome. Just like the spare tire required forward thinking from the human engineers that designed the car, the forward thinking mechanisms in the human genome and the genome of living organisms required a forward thinker, which points us right back to God, the God of the Bible. So gene regulation regulates where, when, and how gene products are expressed. And going from DNA to a functional product like an RNA product or a protein product is gene expression. And it turns out that much of the genome that was assumed to be junk is directly involved in gene regulation and gene expression. So it's we're looking at multiple overlapping codes. The information is nested and it's functional on so many levels. And I could do a whole presentation on that. So I'll skip ahead a little bit for sake of time. So notice this when it comes to endogenous retroviruses. And this is a paper from 2021. So every single year, more and more evidence is coming out on ERV function. So notice this, switching sides, how endogenous retroviruses protect us from viral infections. Long disregarded as junk DNA. What I find fascinating is a lot of the times you'll read these papers and it almost seems like it's an intelligent design or a creationist paper. I mean, they're admitting right here in this paper on endogenous retroviruses that long disregarded as junk DNA. So yeah, they assumed that these ERVs were junk, useless, evolutionary leftovers, or as they put it, genomic dark matter. Endogenous retroviruses have turned out to represent what? Important components of the antiviral immune response. They not only regulate cellular immune activation, but may even directly target invading pathogens. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. So this slide kind of makes me laugh. Cool story, bro. So how did the evolutionists attempt to address the evidence for ERV function? Well, they'll say, well, you know, these functions in embryological development, determining cell types, gene expression, gene regulation, the antiviral roles that they play, you know, they were co-opted with no evidence though, because you'll say, okay, if they were co-opted or these functions were gained somehow through evolutionary processes, show me one single technical paper, show me one single example of a non-functional endogenous retroviral-like element going from non-functional to functional. And they can never provide you that data. And there's a really militant evolutionist by the name of Dr. Dan Stern Cardinal. He's a virologist, and you know, he's got a career fighting young Earth creationists. And he recently admitted, I would say about a month ago, in a show that he was doing where he was attempting to refute my book here. And he was forced to admit that no, we've never observed in real time a non-functional endogenous retrovirus going from non-functional to functional. And so these arguments that the evolutionists put forth in order to save the day for evolution, they're rescue devices. And what I say is, you know, cool story, bro, because what we get is a lot of imagination, right? Evolutionists hope, dream, and imagine that whales and pine trees are related through common ancestry. I was talking about this earlier, nested hierarchies. And I'll get my mouse here to kind of show everybody. In its most basic forms, you know, a human shares more with a chimpanzee than a human does with a lizard, or a human can be grouped with a chimpanzee in terms of, you know, being a primate, if you want to look to taxonomy, and they share more than a salamander or fish. Is it any shock that if these ERV sequences are created units of DNA function, if we share them, okay, I wouldn't be surprised if the entire biological world has endogenous retroviruses, since they are necessary to sustain healthy life processes in the cell. Therefore, yes, we would predict, of course, or assume that a human and a chimpanzee are going to share more of these ERV elements than a human and a lizard or a mouse. This is just basic because we share more anyways in terms of anatomy and morphology with the chimpanzee than the mouse. Okay, humans and chimpanzees, they both have binocular vision. They both have a lot of similarities that they wouldn't share with a mouse or a lizard. And this goes right back to the design hypothesis, because it turns out human engineers build in both homologous patterns and also nested hierarchical patterns. Why does a sedan share more with an SUV than both a sedan and an SUV share with, let's say, an airplane or a bicycle? This is just hallmarks of design. And humans accidentally build in nested hierarchical patterns. Again, humans build in homologous patterns. All types of vehicles from all manufacturers, they you know, build with these similarities. Windshields, tires, same kinds of rubber. A lot of the sedans built from a certain height from the ground. Similarities just demonstrate common design. So notice this paper right here. This is a fascinating one that goes over the embryological development. So as we have summarized in this review, HERV, so again, when you see an H there, that stands for human. Human endogenous retroviruses appear to play important roles in physiology, fetal development, and human evolution. If the accidental infection of a mammalian ancestor by an exogenous retrovirus had never occurred, I want you to pay close attention to this. The placenta and the mammals that produce it, including humans, would have never existed. What's that saying? That's saying without endogenous retroviruses, we couldn't exist. I don't know about you, but I like existing. And I think some of the evolutionists like existing. And we require these endogenous retroviruses to exist. So yes, we are going to share them with the rest of the biological world, because the rest of the biological world also requires these ERV sequences to exist. Existing is important. So the fact that we share them is not evidence for common descent. And I'm going to go over a few of these papers, but in my book, and I highly recommend it, because I want to keep this under an hour, and time's flying by. So in this book, I've got just paper after paper. If you go to the last chapter that absolutely dismantles endogenous retroviruses as evidence for evolution, you're going to find dozens and dozens of papers that you can throw at these evolutionists. And a lot of them, I've debated a lot of them, and they're not even aware of the endogenous retroviral function. And they're thrown off. And then they can't provide you a paper, and they assume, well, maybe there's a paper out there that shows these endogenous retroviruses becoming functional. But then you got PhD scientists like Dr. Dan Stern-Cardinal, who's militant against young Earth creationists, admitting that no paper exists like that. And that's it. All they have is storytelling, imagination, and these arguments don't work. I'm sorry. So endogenous retroviruses have recently been suggested as mediators of normal biological processes, such as what? Cellular differentiation and regulation of gene expression. One thing I want to point out in terms of this genome-wide activity, okay, that we've discovered, is that with all this amazing of activity, evidence for activity, the evolutionists, they want to say, well, this activity is there, sure, but it's spurious, it's noise, it's not really doing anything. That doesn't make any sense, because if this activity was not useful, or the transcription was simply noise or spurious, then guess what? There's something called natural selection that should have weeded out this genetic baggage millions of years ago. Okay, this would be incredibly wasteful of resources and energy for the cell. And this is a strong indication that this activity is in fact useful and beneficial and serves an important role, and not spurious and just noise like these evolutionists want to say. Again, that's just another rescue device. And these endogenous retroviruses exist within this activity, which says that they're mostly active as well. So, and the other question, which is answered by a couple of the papers that I've gone over so far, that talk about their amazing functionality in terms of not only determining cell types, gene regulation, but in fighting off viruses. And they oftentimes do this through viral mimicry. Okay, so the question is that the evolutionists have is, why do ERV sequences resemble viral genetic material, as I covered in the details portion of this presentation? Well, it turns out that one of the major functional responsibility of these viral-like elements is that they operate in the innate immune system. They perform an antiviral role. Think of your antiviral programs on your computer. Those are beneficial. Without them, your computer could be invaded by viruses really quickly and break down. And that's the same thing with the human genome and the genomes of the various biological organisms. God front-loaded them with these important DNA elements that can fight off viral invaders. And so they are valuable DNA elements that work considerably in the immune system of their host. And the way that they systematically exercise, I want you to pay close attention to this guy that destroys endogenous retroviruses as evidence for evolution. The way that they systematically exercise their antiviral effect has to do with their sequence resemblances to viral material. They require their particular structure, the LTRs, the GAG, the POL, the ENV gene, that they say, well, these are so similar and they nest within exogenous retroviruses. Yeah, that is required. And their function necessitates their specific structure, okay, because the way that they exercise their functional roles depends on their similarities to viral material. And so without the nature of ERV sequences, the way that they're designed, they could not do the significant job that God designed them to perform. The reason why God front-loaded most of them in the beginning, given the design diversity hypothesis. So we should be thanking God that they resemble viruses, This isn't bad design. This is not evidence for evolution. Their functional roles depend on these similarities. And just paper after paper that I could do probably a 10-hour presentation. So it's not just one or two papers, because another response these evolutionists will put forth all the time, well, it's one or two functions. No, it's not one or two functions. It's dozens and dozens of papers in the technical literature that talks about function after function, genome-wide functionality and activity. Again, here's three more papers. Retroviral promoters in the human genome. This has been known for a long time. This one's 2008. Earlier, I went over a paper that was 2021. I've got papers from 2022. This has been well known, and the evolutionists have to update themselves. These data suggest that ERVs may regulate human transcription on a large scale. And again, the evolutionary community, they've completely dodged explaining how these functional roles can come about. And you can, pause the screen and check these out for yourself. Scientists identify new beneficial function of endogenous retroviruses in what? In immune response. In immune response. And another thing I want to point out is we understand that a lot of non-coding RNA genes, like pseudogenes, but also some of these DNA elements like ERV sequences, they only function under certain circumstances and certain windows. And what I mean by certain windows is certain developmental windows. So you have a lot of these ERV sequences that are functional during the various windows of embryological development, where they assist in cell differentiation, cell migration. But when they're done performing their role, they're shut off or suppressed. So they're no longer performing their job because they already have. Think of a big construction building where you have construction workers that are building the building. But once the building is done and set and ready to go, well, those construction workers leave and they go to another job. Well, those construction workers were working on the building, the construction site, and now they're done. They've moved on. And a lot of ERV sequences do the same thing. They function in the embryo. Once they're done their job, they're suppressed or they're turned off. But the evolutionists, they want to look to a lot of these ERV sequences that aren't really doing anything during the life of the organism and assume they're junk. When the reality is, no, they're turned off or they're suppressed because they've already done their job. And, here's papers that go into things like that. Endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. Remember, if it wasn't for these ERV sequences, we wouldn't exist. We wouldn't exist. Then there's something called shared mutations. So they'll try and point to what appears to be shared mutations within the ERV sequences themselves. Firstly, I need to point out the major difference between both the creation and the evolutionary model. Creationists explain the vast majority of genetic diversity as being the result of creation, as I pointed out earlier. That's why I wanted to set the foundation. Again, created heterozygosity. Heterozygosity just means a state of DNA diversity. But the evolutionary community, they explain all DNA variation, all DNA diversity as being the result of what? Mutations over time. So where they look at a genome, where they look at, let's say, a DNA letter at a DNA position, they're assuming it's a mutation, when in fact it could reflect design. It doesn't necessarily have to be a mutation. That's an evolutionary-based assumption. And what we're understanding in the human genome is like putting a kid who's 5 years old underneath the hood of the car and telling him to remove anything that has no use. He's going to start pulling things out left and right because he doesn't understand the function of the car. And that's what we see in the human genome. It would be like myself. I speak no Russian. And I go over to Russia and they hire me as a proofreader, a Russian proofreader for their books. I'm not going to know what the difference is between a spelling. I'm not going to be able to identify spelling mistakes because I can't speak the language. And evolutionary scientists, those studying the genome, they are in the infancy of understanding the DNA language. So where they want to say it's a shared mutation, a lot of times, especially with the pseudogenes, then we figure out that these aren't mutations. Okay, these are assumed to be mutations when they're actually reflection of initial design. And I've got a bunch of slides on this as well. But even in the case of real shared mutations, it's no help to evolution because we understand that the mutation rate, and there's a concept called genetic entropy. Low impact, nearly neutral, deleterious mutation accumulation. We as humans, we inherit roughly 100 new mutations per person per generation. Okay, that means we're consistently getting more every single human being on this planet is multiply mutant. And since the mutation rate is very high in the genome, in populations, that means a lot more mutations are going to hit various spots in the genome that evolutionists once assumed were maybe not what's called a mutational hotspot. Positions in the genome that are more susceptible to being affected by mutations. Okay, and you can see the same thing with vehicles. I mean, you could just Google, you know, which part of your vehicle is most susceptible to damage. And you're going to find that your engine's very susceptible to damage. There's certain parts of the car that's more susceptible to rust. So if you have two different cars and they got rust marks on the exact same spots, well, does that mean that they inherited those rust marks from a common sedan ancestor in the Arctic? No, common problems due to common environment. And when it comes to the genome, since what we know is it's a lot more functional than the evolutionists have ever assumed, there's what's called mutational hotspots in the genome where the mutations, a lot of mutations that are accumulating from generation to generation, they can hit certain spots in independent lineages. So a chimpanzee might be hit with mutations in the same spot that a human is, not that they inherited those, just the fact that there's similar mutational hotspots. And we can see that with the ERV sequences. So here's a number of papers. Again, everything I say is backed up by the scientific literature. Okay, non-random mutations and hotspots. And I've got a whole section of this on my book. This is an argument I deal with in depth. And there's a lot in the literature on this. Okay, unexpected findings on mutations. Notice this. It turns out that mutation is a very non-random, and it's non-random in a way that benefits the plant. So this is a paper that goes over how a lot of these mutations that have occurred have occurred non-randomly, which means they can occur in similar spots. Again, non-random mutations here. I've got a ton of papers on this. Notice this. Mutations are usually thought to be so rare that when we see the same mutation in different individuals, the assumption is that those individuals shared an ancestor who passed the mutation on to both. But it's possible that the mutation rate is so high. Remember, I pointed that out. The mutation rate is now so high. The genome is mostly functional. That means the mutations that hit every single generation are going to be, by definition, deleterious, damaging, disease-causing, and harmful. That means species have what's called shelf lives. Okay, if species cannot persist for millions of years in the future, species could not have persisted for millions of years into the past. Mutation accumulation alone destroys both evolution. It's a double whammy. Destroys both evolution and deep time and an old Earth. And if the mutation rate is so high in some of these places within the genomes, then guess what? Just as this paper concludes, these mutations could occur independently in several different individuals. So these ERV sequences that you find, let's say in chimpanzees and humans, and you have an ERV sequence that functions in determining cell types, or they have a role in embryological development, well, there also might be hot spots within those ERV elements that are more susceptible to the same mutation in the same way that cars from different manufacturing companies are susceptible to the same type of damage in the engine or the trunk of the car or rust spots. It doesn't demonstrate shared ancestry. And again, the differentiating line of evidence is function, and function is on the side of the creationists, and the evolutionists are incapable of addressing these arguments from function. So as I start winding down here, I do want to point out that, again, when it comes to the concept of genetic entropy, and also I want to point out the fact that natural selection it's a fine-tuning mechanism that keeps a species as strong as it can be. Natural selection actually can't see these deleterious mutations because they only have very subtle effects on fitness. They're harmful, but they're not harmful enough to kill an organism. Okay, if a zebra is born with one leg that's shorter than the rest, then yes, that mutation is probably going to be enough to kill the organism because that's the zebra that's going to be dinner for the lions. Okay, but a lot of mutations that accumulate, they're harmful, but they're only slightly harmful, like a single rust molecule on a car. Okay, they're going to build up unnoticeably, but eventually your car is going to rust out and you're going to have to get a new one. Or a single spelling mistake in a book the size of an encyclopedia. On its own, the single spelling mistakes are inconsequential, but over time they build up And as they build up, the message in the book becomes more and more garbled and unreadable. So here's a paper, our future genetic load, the human germline mutation rate. An average newborn contains what? 100 de novo mutations. 100 slightly deleterious mutations. Under the present model, effectively neutral. So it's not accurate when the evolutionists want to say, well, these are completely neutral, which means they're not damaging. And so they can build up and act as a hidden reservoir of future evolutionary change. That's essentially the neutral theory, the theory of neutral evolution. No, they're effectively neutral. And notice this. This is evolutionary population geneticists admitting this. The rate of loss of fitness per generation may amount to 10 to the 7 per generation. And it talks about how over time, species will get less and less fit. It seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. This is what I've been saying. All mutations must have some effect, even if that effect is vanishingly small. So we have a high mutation rate and we have a genome that's mostly functional. And so these ERV sequences are therefore going to be susceptible and prone to the same kind of mutations in independent lineages. And so if you find a similar mutation in a human and a chimpanzee ERV sequence that functions in the embryo, it's not evidence that that is a mutation that was inherited from a common ancestor. No, that argument just doesn't work. Okay. And another argument that they'll say, like the Dr. Dan Stern cardinals of the world, okay, these guys that are militantly anti-YEC, they'll say, okay, well, we've got these functions, but they were expected. Oh, really? Just read a few sentences from these papers. Here's one from 2013. Notice this. Recent studies also point towards an until now expected, nope, unexpected role of the immune system. These weren't expected. These weren't predicted. They were unexpected. So the evolutionists have to save face because their favorite argument is endogenous retroviruses, but endogenous retroviruses have been absolutely decimated by their own scientific literature. Unexpected role of the immune system for the control of these elements. Again, unexpected functions. Unbiased analyses of these genomic maps have uncovered endogenous retroviruses as an unexpected ally in the regulation of the human immune system. Think about this, guys. It's funny. It's humorous. So apparently these exogenous retroviruses invaded millions of years ago. They invaded, the cell. And then they invaded in just the right way where they can be passed on vertically through the germ cell or reproductive cell lines. And then they decided to evolve the ability to fight off what they used to be. That's ridiculous. That's like a robber breaking into my house to steal things and cause harm. But then realizing when he gets into the house, you know what? This couch is nice. The TV is nice. I think I'm going to move in and benefit this house rather than rob it. It's ridiculous. There's a lot of fairy tale imagination storytelling going on. Look, paper after paper. I'm going to go over a couple of these and then wind it down. I think I'm coming at the hour mark. So although TEs, when you see TE, that stands for transposable element. In general, been regarded as non-functional junk. So they admit all throughout the technical literature that, yes, we used to think that these were junk DNA. We used to think that they were junk, but now we understand they're functional. But yet you still have some of these evolutionists in the YouTube world trying to say that these were expected. We never assumed that they were junk. Their own literature refutes them. They're arguing against their own literature that they want to hold up and praise. The scientific literature, you hear it all the time, peer review, that's the holy grail to them. Well, their own secular peer-reviewed literature refutes them. Long been regarded as non-functional junk or selfish DNA, parasitic elements, or harmful mutagens. Studies have revealed that TEs, notice this, have a substantial and sometimes beneficial impact on host genomes in several ways. Paper here. Transposable elements are a prolific, it's not just one or two, prolific source of tightly regulated, biochemically active. One single ERV sequence is more complicated than your most complicated car out there, the space shuttle. They can't explain the complexity of these ERV sequences that are involved in determining cell types, as we've pointed out, involved in the immune system, involved in embryological development, to the point where they admit if we didn't have them, we wouldn't exist. They're not evidence for evolution, guys. They're not evidence for evolution. ERVs appear to play important roles in physiology, fetal development, and human evolution. Keep going. Our results suggest that a subset of Ts are important for gene regulation in early mouse development. Okay, so they've done experiments where with the mouse genome, it's developing, and it turns out that there's a specific class of the specific retrotransposon that if you turn it off or snip it out in the lab, the mouse is developing, but you turn it off or snip it out and it stops developing, it dies, because the mouse requires this retrotransposon to develop and live in the first place. Again, it's not evidence for evolution. It's evidence for design, amazing design. The Bible says that they will be willingly ignorant. In the last day, there will be scoffers denying the creation, the worldwide flood, the coming judgment. They want so badly to believe that pine trees and whales are related. Our results suggest that TE insertions increase the regulatory potential of the genome. And this is just a very tiny fraction of what I have in this book. Again, I could go on for 5 hours. This is just a tiny, all you need is one. All you need is one of these 100 papers to destroy endogenous retroviruses, because guess what? Even their best, quote unquote, their best, still believes that whales and pine trees are related, so I'm not impressed. But even their best admits that, no, we've never seen this function gained. I want you guys to look at this. Overall, in all the sources I have listed here, so you can look up the papers yourselves if you want. Overall, our results provide direct evidence for retrotransposons in actively shaping cell type and species-specific chromatin genome architecture. But no, apparently these are just the ancient remnants of past viral infections. No, it doesn't work. And here's 2 more papers. For sake of time, I won't go over those. But I do like this one from this paper. Far from being junk DNA. So again, admitting that they once assumed they were junk DNA, the pervasive retrotransposons that populate the genome have a what? Have a powerful capacity to influence genes and chromatin. Again, ERVs are not evidence for evolution. Here's another paper, Emerging Roles of Endogenous Retroviruses in Immune Epigenetic Regulation. We have this whole extra layer of complicated information called the epigenome. And we've got this, it's a control system, because if every single gene was turned on, that'd be bad. If every single one of your genes was turned on at the same time, that would lead to disease. It'd be harmful. You'd die. So we have to have some kind of control mechanism and control system. And that's exactly what we have in terms of the genome. We have this control system, which again points us back to the forward thinker. And even the specific elements that we find, because not every single endogenous retrovirus in the genome is found in its completeness, you could say, where you've got the LTR, GAG, POL, ENV. Now you find what's called a lot of solo LTRs. LTR stands for long terminal repeats. And it turns out that those solo LTRs are just functional stretches of DNA. Here's a couple papers here, how they function in human genome, acting as promoters. Just functional stretches of DNA. So as I wind it down here, I want to recommend a few other books from our ministry. The Independent Origins Handbook, Refuting the Critics, all their best arguments. We've got books refuting those arguments in great detail. They're comprehensive. And one argument, again, when it comes to this co-option, okay, just think about it, guys. If viral DNA is randomly inserted into the genome of a living organism, is that viral DNA likely to improve the organism? No, that viral DNA is far more likely to be damaging to the cell. That viral DNA is not going to improve genetic functionality. It's not going to suddenly be functional in the immune system, in the embryo, to the point where we now require them. No, that's fairy tale. It's not science. We believe in science here. Okay, it's not going to evolve that ability. And the best analogy or illustration I could point out is if I were painting a picture and I decided to close my eyes for portions of it, I would more than likely ruin the painting. If I'm just applying random brush strokes blindfolded, no, I'm going to more likely ruin the painting, not make it better. And it's the same thing in genetics. It's much easier to destroy a gene than to make it better, or to build one entirely, which is what evolutionary theory requires. So it's much easier to disrupt genetic functionality than to make an entirely new function. Or what they'll say is, well, the ERV altered its pre-existing function. No, that's not going to work either. Okay, they've never seen the altering of a pre-existing ability of the exogenous retrovirus to the point where it's now necessary and essential to sustain healthy life processes in the cell. So I'm sorry, these are all rescue devices. We've dealt with them in a number of our books. These are the two current books I'm working on now. So an expanded edition of Special Creation, which goes into the amazing evidence, scientific evidence for Adam and Eve, a literal Adam and Eve, and the Y chromosome, the mitochondrial DNA, mutation rate, so on and so forth. And then I'm also working on the Human Evolution Handbook, where I address all of their favorite so-called hominids and transitional forms, like Australopithecus afarensis, Sediba, Habilis, Floresiensis, Erectus, Neanderthalensis, so on and so forth. So hoping for that to be out in the next several months. And then 3 must-have books unrelated to our ministry that I would highly recommend, that just absolutely dismantles evolutionary theory that I believe every single young earth creationist should have in their library, is Genetic Entropy. We talked a lot about today about genetic entropy, mutation accumulation, and the fact that most mutations, they're too subtle to be selected for. So selection can't see them. So they build up, they're subject to genetic drift. And what's fascinating about the reality of genetic entropy is if we take this point of most accumulating genetic damage and we take it back to a point of least accumulating genetic damage, or just no genetic load, that's a point of perfection. That's a point of increased longevity, like we see in Genesis, where our biblical patriarchs were living in hundreds of years old. That points us back to Adam and Eve that had no mutations. They had a perfect genome, living in a perfect world, essentially. Contested Bones, written by my brother here, Christopher Roop, who's been a huge blessing. Dr. John Sanford, same author as Genetic Entropy. That is the most up-to-date book debunking human evolution. You'll find comprehensive beatdowns of all of the evolutionists' favorite arguments, including some of their genetic-related arguments, like chromosome 2 fusion. Turns out that the so-called fusion site represents an active promoter inside a human gene. I'm sorry, evolutionists, but you don't get an essential gene, a functional gene, by slamming together two chromosomes. No, this so-called fusion site is overlapped by a functional gene. And the so-called fusion site is not a fusion site. There is no fusion. It's actually a promoter for that gene. And Ken Miller, militant evolutionist, he said if there was no fusion, there was no evolution. I know this has been long and technical, but I want to repeat that. If there was no fusion, there was no evolution, because chimpanzees have 48 chromosomes. We have 46. They require a fusion to explain those differences. And it turns out that this fusion, which they say is a fulfilled prediction, no, it was a prediction that turned out to be wrong, because it's not a fusion. And the last book I recommend is Replacing Darwin by Nathaniel Jeanson. The New Origin of Species. So he goes over some fascinating predictions and a research program he's been doing, demonstrating how humans only go back a few thousand years, and how he can detect genetic signatures and stamps in the Y chromosome, and hopefully in the future the mitochondrial DNA, where humanity only goes back roughly 4,500 years to what? Noah and his family, Y chromosome Noah. So that's fascinating. And that's it. So hopefully I wasn't too long. Oh, there we go. Exactly an hour. So I actually cut a lot of it down just to kind of wrap it up. So that's the last thing I want to say. That's why I highly recommend the book. This was just an hour, and it only represents maybe a quarter of the book. There is a ton here that I want. Any argument that these evolutionists can put forth, whether it's the nested hierarchies, whether it's the rescue devices when it comes to the ERV function, it's all addressed in here. So this is a very comprehensive source. I'll wrap it up there. Thanks, brother.
Wonderful, wonderful. Yeah, that was definitely a slam dunk presentation. Do you got just a few minutes for a few questions there, Donny?
Sure.
All right, we got one that came in. It says, are ERVs mutating or breaking? If they are, wouldn't that prove that they were better and more functional in the past and breaking down over time?
Yeah, that's a very good question. And this goes back to the concept of genetic entropy. So when God would have initially created Adam and Eve, for example, okay, and he would have front-loaded Adam and Eve. This applies to the genomic archetypes. Actually, what this is called in technical terms is the baronome, right? Baromenology. The baronome would consist of all the functional DNA elements in genes in a baramen. Okay. And so what this would indicate is the original created ERV sequences, over time, they would be breaking down. They started off functional, doing their job right, but as mutations have accumulated, right, the fall, the curse, that they have been breaking down, and they've been breaking down in separate lineages. So if we find similar mutations in different lineages, let's say humans and chimpanzees, that doesn't mean that we inherited those mistakes or mutations from a common ancestor. That just means that both lineages are experiencing the same type of degeneration related effect. So good question.
Awesome. Thank you for that. And then another one came in. It says, if ERVs are junk DNA, does the body supply energy to them? If so, wouldn't that refute that they're worthless and that the body is not using them?
Right, exactly. There's a lot of activity going on in the genome, and the endogenous retroviruses are very active. It requires a lot of energy, okay, to add a single nucleotide to a growing RNA chain. A lot of energy. It's an energy-intensive process. It's an energy-intensive process to transcribe all of these DNA elements. So it makes no sense that this activity would be spurious or just noise. The activity actually suggests function. That's why a lot of creationists have made very specific predictions on function. And in my book, actually, I have a whole section where I make some novel predictions on fixed ERV sequences versus non-fixed and function, so on and so forth. So if you want to see more details on that, again, I recommend the book.
Awesome. And then there's another question that came in. This one's from Raw Matt. It says, Donnie, did evolution predict function in ERVs? And did YEC predict that there is not junk DNA?
Right, exactly. So you'll see throughout my presentation in a number of technical papers that the evolution community admitted that, yes, we once assumed that these ERV sequences were junk. Now, in the junk DNA in total, as I pointed out in my presentation, now ENCODE revealed that, you know, between 60 and 80% of the genome was active to some extent. They may have expected one or two functions, but they definitely did not expect the number of functions that have been found in the junk DNA. So no, they didn't predict, and they still refuse to make predictions. Because again, Matt, as you know, they require genomes of junk because mutations are accumulating, and the evolutionist needs the majority of the genome to be junk in order that those mutations that are accumulating in what's called junk or neutral regions. But if those mutations are accumulating in functional regions, that means degeneration is inevitable and evolution is impossible. So great question.
Great answer. And then the last question that we have here is this one. It says, have knockout tests ever been done removing ERVs? And if so, what were the results? Perhaps a prediction can be made on this.
Fantastic question. The gold standard of genetic function-related testing is genetic knockouts. For humans, as you can imagine, that's unethical. Right. Well, you know what? Some of these evolutionists like Richard Dawkins, they believe that the majority of the genome is junk. So maybe volunteer yourself to have massive portions of your genome knocked out, and let's see how you survive and feel after that. But we have done it a lot on mice. And one thing I want to point out is, and something I pointed out in the presentation, that a lot of these non-coding RNA genes like pseudogenes, but also these DNA elements like ERV sequences, they're only active and functional in certain periods of an organism's life, like these developmental windows. Then they're shut off. So if you were to knock one of those out after they're functional, or maybe they're not turned on via environment, then it might not have any effect. But that doesn't prove that they're not functional. It's just they're not being called upon to do their job, or the job that they had, it's done, right? Well, once you're born, your embryological development stages are over. So whatever genes or DNA elements were involved in those stages, they're done. They're suppressed. And they might be turned off and later called upon due to adaptive episodes, environment. And so, but yes. And I pointed out one example where you have this specific class of retrotransposon, Matt, in the mouse genome, where the mouse is developing, and then you snip out this retrotransposon. Well, the mouse is developing. Once you snip it out or turn it off, guess what? The mouse dies, because the mouse needs its development requires that retrotransposon. So if you snip it out, it dies. That's not evidence for evolution. I'm sorry. I mean, Matt, you like to exist. I like to exist. And a reason why we can exist is the existence of these retrotransposons. So very good question. Appreciate it.
And if any of you atheists out there want to take the challenge of debating Donny on any of these matters, go for it. I'd like to see your arguments. I'd like to see how you present them. And we could even do it on my channel, Matt Powell Official, just for the kick of it. And we could do a live debate over there. Donny, is that something that you'd be up to doing?
Absolutely. Absolutely. I've got a few debates actually scheduled for the next couple months. I have a debate on endogenous retroviruses with Luca Madugno. He's read my book. He says he's making lots of notes. So it's going to be an interesting debate. And, I don't know what he's going to do with the fact that Dr. Dan Stern Cardinal, right, their fearless leader, has admitted that no, they've never seen, and I'll say they never will, see a non-functional endogenous retrovirus going from non-function to function. And not only that, but going from non-function to something extremely functional and critically functional in what, Matt? Embryological development, determining cell types, the immune response. So I do have some debates scheduled already, and I'll take on any evolutionist when it comes to the topic of ancestry. So absolutely, brother.
Awesome. Awesome. Donny, thank you so much for coming on. Thank you for taking the questions. And then is there any closing thoughts or any closing remarks that you want to share with the audience before we close out?
Good question. I appreciate you having me here, brother. And, I just encourage people to share this around. And anytime these evolutionists bring up endogenous retroviruses as evidence for evolution, send them my book, send them this lecture. And I just want people to know that it's 2022 and it's a great time to be a young earth creationist. It's a great time to be a young earth creationist. The evidence on our side is overwhelming, and our faith, we can be strong. Our faith can be strong in the word of God. And the word of God claims to be the history book of the universe, and its claims have been validated using empirical scientific data. So yeah, I think that's it for final words, brother. I appreciate it.
Amen and amen.
The layout of the suite of viruses, viral remnants, is exactly the same and could not happen unless it was inherited by a common ancestor. And so that's where we're at. So
Herbs, like when you do the phylogeny of herb sequences, they nest within retroviral sequences. They have sequences like both the end regions and the genes that are only found in retroviruses, and they can actually be expressed in the human genome. Like some of the genes from these dead viruses can be expressed and cause problems. It's just all the evidence just says like they're from retroviruses, and that's the end of it.
Accordingly, retroviruses could have originated from within the host genome. Okay, you admitted that the host would have predated the virus. Okay, that's exactly what the variation-inducing genetic element hypothesis put forth by creationists suggests. Okay, accordingly, retroviruses could have originated from within the host genome instead of invading it from outside. This had to have been the case in any scenario since viruses are dependent upon host cells. This means that the cell's genome had to predate the viruses. You can read all this yourself.
So going back to the question I asked, is there a way to differentiate a virus mimic and a legitimate ERV?
Right, so that's a good question. I would say the majority of what we're looking at in terms of these quote unquote genomic fossils that evolutionists such as yourself look to and compare across different groups, different species that fall within a nested hierarchical pattern, as well as the mutations that occur in these LTRs, these LTR elements. I would say that the vast majority, if not all of those, are created units of DNA function. Endogenous Retrovirus Handbook. So for anybody who is interested, this is a topic that is frequently discussed these days. You can get the full color version. Benjamin did a fantastic job. Probably the most comprehensive book you're going to find on the market on this specific topic, black and white version. And you won't regret it. You won't regret it.